Hey, well, thank you. And first I wanted to start by expressing my appreciation for being involved in this discussion as a microbiologist living and working in this area for the past 27 years. We've taken great advantage of the IOM forums and symposia that have been put on and have been fantastic and shaped our thinking in a number of ways. So I've been here, I'm here to give a regulatory perspective, and to give that, I think I need to give you a little bit of information about the point from which I have that perspective. You went through my entire institutional address. I'd forgotten that I put that down. But I'm in the laboratory of enteric and sexually transmitted diseases, and I think we're notable, and the reason I'm here is that we receive, among others, IND applications involving fecal transplant, probiotics, and phage therapy. A few things I want to mention just so you can understand where I'm coming from. CBER follows the research reviewer paradigm, so that means that at least some of the people reviewing these files have active research programs. They're bench scientists. The second important thing to note is that we don't do that alone. The bench researchers tend to be product reviewers. They tend to review the CMC or chemistry, manufacturing, and control information. But we work together with clinicians, statisticians, toxicologists, and so forth on any given file. Over the years, my regulatory experience has involved mostly enteric diseases, vaccines, as you might expect. But in recent years, some of these issues that we're here today to discuss have come to the fore, and I just wanted to spend a little time going over each of those. And those are live biotherapeutic products, which include as a subset probiotics, fecal transplant, or FMT, and phage therapy. So I'll start with the live biotherapeutic products. This is an FDA term. Basically, it means the same as probiotics, but we prefer it as a definition. And we get these INDs to review when that, very often a probiotic off the shelf, but is being used in a clinical trial for a specific disease indication. And that's the point legally where it becomes a drug, also a biologic, but a drug, and that's where we review it and where we require an IND. Now, especially in the early phases of the IND process, safety is a paramount concern. Probiotics, people think, are generally considered safe, and I'm not saying they're not, but we have to review them in a safety context that's a little bit different. Often these clinical trials are being used to administer this to highly vulnerable populations, premature infants, infants with NEC, immunocompromised people, and so on. And so the bar for characterization of these products it goes up a little bit. And all I'll say from a product standpoint is that one of the primary indicators of safety for a probiotic preparation is microbial purity. And that can be challenging because these are products that contain 10 to the 9th or 10 to the 10th product organisms. Finding contaminants or other pathogenic bacteria can be difficult. So I'll move on quickly to fecal transplant. Clearly of great interest here today, and generally speaking. It's being investigated, as has been said many times, for treatment for recurrent or refractory C. difficile infection, inflammatory bowel disease, obesity, and interestingly to me, autism. Again, because it's being used to treat a specific disease or being investigated in that regard, it's a drug and requires an IND. Now with fecal transplant, I think we can put the safety concerns into two large categories. One is the inadvertent transmission of disease, and the other is what are the long-term safety concerns associated with a change in the microbiome. I mean, we've heard today, and we've all heard how potent a force this is in shaping our biology and our metabolism. it stands to reason that not all of those changes may always be beneficial. And so that's something that we try to keep in mind, although I would agree with your characterization that to date the safety profile for FMT, certainly in the context of C. diff infection, seems to be good. So to begin to address this concern, and we've really been in the FMT regulatory business only for a couple of years now, we had a public workshop. The FDA and NIH together sponsored a public workshop last year. We brought together bench researchers, clinicians, and regulatory personnel such as myself. And it was a 2-day meeting. I thought it was extremely good. I got a lot of positive feedback. I think we had a lot of frank and fruitful discussion. One of the things that came out of that meeting was that we expressed what had always been our regulatory standpoint, and that is that an IND was required to do FMT. In the wake of that workshop, there was much discussion, reaction, and so on, and we very quickly went to a stance of enforcement discretion for FMT when used for C. difficile infection, and that's currently the situation. We are in the process of revising that short guidance, but that is currently in effect. And the other thing that I really took away from the meeting is that we really need a lot more research into FMT and the microbiome to begin to regulate this more effectively. One, in terms of mechanism, looking at product as to whether it can be predicted to be successful, and also in terms of safety. And I see I'm running out of time, so I'll only very quickly touch on phage therapy, but I thought it was very interesting the way Dr. Riley brought up bacteriocins this morning, and phage therapy are sort of similar. They're kind of the ultimate narrow spectrum anti-infective, and they have a high presumption of safety due to their essential lack of interaction with